Okay, welcome back. This is the second lecture in our set of 2 for Chapter 15, Genes and Proteins. Just want to remind you, and always as we think about transcription and translation, always keep in mind the central dogma and the fact that information travels in one direction from DNA, double-stranded, to single-stranded RNA. That's your message. The process is transcription. The DNA gets transcribed into the message, and then the message gets translated into the protein. So we in the last lecture we got to the point where we have made the messenger RNA. Now, remember that the messenger RNA is transcribed from DNA in the nucleus, but before it can be translated into protein, it must be modified. In other words, it gets translated into protein outside in the cytoplasm, but before it can do that, it needs to get modified so that it doesn't get torn up, trashed out, seen as something foreign, and dissolved, digested by enzymes in the cytoplasm. So we need to process it, and so that's the idea behind making pretty before the party. The party is when the message gets translated in the protein. Okay, so what happens before we can get to translation? After the message or RNA has been transcribed, we call that the primary transcript. It has to get modified. Now, there are two terms here: post-transcriptional modifications or pre-translational modifications. Means the same thing. I don't really care that you remember either term, but the point here is the modifications are made after the primary transcript is created, post-transcriptional, or before the message is translated, pre-translational. Okay, and these modifications occur prior to the message leaving the nucleus. All right, so spliceosomes, we have enzymes here called spliceosomes, edit the message. So as we'll see in the next picture, this message includes chunks of DNA, chunks of nucleotides that code for specific proteins, and then it has chunks of nucleotides that code for nothing. The chunks that code for something, we call them exons, E-X-O-N-S, and the chunks that are useless and code for nothing, we call those introns, I-N-T-R-O-N-S. So part of the modification is these spliceosomes come in and they remove introns, which are non-coding, and they splice together, spliceosomes, they splice together exons, which are going to remain. Those are the coded parts. Those are the parts that make the protein. All right, and the other modifications are the addition of a 5' cap and a 3' poly A tail. What does all that mean? Well, remember, there is a polarity to the message. Just like there's a polarity to DNA, 5' to 3', there is a polarity to the messenger RNA, to the message, 5' to 3'. So that this thing doesn't get chewed up by lysosomes in the cytoplasm, we need to modify it. And the one way that we modify, or the cell modifies the message, is on the 5' end. It hooks this methyl group, and this it's a methylated. Okay, we call it methylated because it has a methyl group on it. And it has a GTP. Now this is just like, or very similar I should say, to ATP, adenosine triphosphate. That's an adenine residue with 3 phosphates attached. This is a GTP. This is a guanine residue with 3 phosphates attached, okay? But it acts the same way. There's energy in the pyrophosphate bonds of the GTP, just like there's lots of energy in ATP. And so later on we will cleave these bonds and use that energy to do other things with regard to translation. Okay, so that goes on the 5' end. It's a methylated, that's a carbon with 3 hydrogens. That's methyl. And that's connected to a GTP. So it's a methylated GTP on the 5' end. We call it the 5' cap. And on the 3' end, we call this the poly A tail. It's just a long string. It could be 5, it could be 10. It's not astronomically long, but it's just a long string of adenine residues. So if you look down here at the 3' end, it's A. All right. And so the process here is polyadenylation, right? It's many adenines attached in series to the 3' end. Okay, so those are the 3 things that happen before the message leaves the nucleus. The introns are removed, the exons are spliced together, the 3' cap, methylated GTP is put on, and the poly A tail, the 3' poly A tail is put on, and then we can leave the nucleus. Okay, so here is the primary transcript, okay, and you can see these purple areas here are introns, the blue areas are exons. So this is the coding part. The blue part is the coding part. The purple part is just trash. We can get rid of it, and that's what happens as the spliceosomes come in. So after transcription, right, these are post-transcriptional or pre-translational modifications. These exons, or these introns in purple, are removed, and the exons here in gold are spliced together. So that's what you see here. Introns are removed, and the 5' cap, the methylated GTP, is put on, and the poly A 3' tail is put on. That's called the mature messenger RNA, and this is the structure that leaves the nucleus. How does it get out of the nucleus? Well, you know this already. We've touched on it a long time ago. You'll recall the anatomy of the nucleus, and the nuclear membrane here has all these little tiny pores in it, these little tiny holes in it. And one of the things that those holes, those pores, are used for is to shuffle mature messenger RNA out of the nucleus into the cytoplasm, where the ribosomes can get to them and start their work. Okay, so that brings us to the genetic code. The ribosomes read the message and they bring in the proper amino acid. That comes by way of the genetic code. The genetic code is just this group of 21 amino acids. You don't have to remember the 21 amino acids. We'll look at this a little bit closer in a minute, in large. You don't have to remember the 3-letter designation for phenylalanine or serine or leucine or tryptophan, okay, TRP. You don't have to remember those. When you do these exercises, you will have a copy of the genetic codes. You don't have to memorize any of this stuff, but you have to have an idea of what's going on. We'll look at it closer in a minute. The genetic code, what you need to know, is universal. In other words, the same genetic code, the same set of 21 amino acids is used to make proteins in humans and in elephants and in bacteria and in mushrooms and in great white sharks and in American toads, whatever the organism, okay? Whatever the organism it is, doesn't matter. The same genetic code, 21 amino acids, is used to make the proteins of every living thing on Earth. That's why it's universal. This is highly conserved. This existed from way back just after the Big Bang, right almost in the very beginning of time, what were these 21 amino acids. And so everything that is alive on the earth today, or that has ever been alive, these 21 amino acids governed the phenotype of all of those organisms, living now or extinct a million years ago. All right, you should also know that the genetic code is degenerate. You've heard this word before. And what that means is some amino acids have more than one codon. A codon is the three-letter designation. That's the three-letter base. Remember, the bases are in DNA, A, T, C, and G, and RNA, A, U, C, and G. Okay, so there's only 4 bases, right? But we have a ton of combinations of those 4 bases, and if you and a codon then is going to be a series of 3 bases. So it's going to be like AUG or CCC or UGC or UAC. Okay, it's going to be like that. That's a codon. It's a 3 base designation for a particular amino acid. And so it's degenerate because most amino acids are coded with more than one codon. We'll get it closer in a minute, but here you can see over here there's 4 codons that code for this single amino acid. Right? The word is degenerate, or a better word I think is redundant. Okay? More than one codon for any single amino acid. Now, a codon is a 3-letter designation for a specific amino acid, and codons are non-ambiguous. Okay, so only AUG is the only codon that codes for methionine. All right, there's no other codon that codes for methionine. There's no other amino acid, I should say, that AUG codes for. AUG only codes for methionine. Okay, so every single codon codes only for one amino acid. Now, you may have an amino acid that has more than one code, but each of those 3 or 4 or 2 codes only for that particular amino acid. So the codons are non-ambiguous. That's what that means. There's 61 codons. There are 3 stop codons. You see them in red over here. I'll show them to you closer in a minute. And there is 1 start codon. And that start codon is AUG, which codes for methionine, and there it is in green. And what that refers to is the fact that as the ribosomes attach to the message, translation of the message into protein cannot happen until that start codon is reached. So you can have a whole series of amino acids, right? But until the ribosome sees that first AUG, that very first one, will tell the ribosome you can start translating now. And so it'll translate AUG into methionine, and then it'll translate CCC, and then UAG, and then GUU, and all the way down the row until it gets to a stop codon. And then when it reaches a stop codon, that codon says, okay, ribosome, you can stop translating now. And then what you get between start and stop, that is the linear protein, or the polypeptide that will eventually become the protein. It's the first level of protein structure, right? So here's the code up close. And again, when you have to do these exercises of translating messages, you don't have to remember all these letters. Okay, they'll be given to you. You'll get a copy just like this of the genetic code. But you'll notice here that you have the first letter over here. Now, remember, the letters are the bases, and the bases for RNA, right, can be U, C, A, or G. There's only 4. And so each codon is 3 letters. So the first letter can be U, C, A, or G. The second letter can be U, C, A, or G. And the third letter, guess what, can be U, C, A, or G. Okay, you can only have those 4 letters, but you can have, a ton of combinations, as you see here. Okay, so here, first letter U, second letter U, third letter U, that gives you UUU, and that's the codon for phenylalanine. The 3-letter designation is PHE. You don't have to remember that. You'll get it. You'll get a copy of the code. But this UUU will only code for phenylalanine. Nowhere on this chart are you going to find UUU coding for anything else. Now, this happens to have another codon that codes for the same amino acid. First letter U, second letter U, third letter C. That also codes for phenylalanine. This UUC and the UUU both code for phenylalanine, but UUU only codes for phenylalanine, UUC only codes for phenylalanine. Here's leucine down here with 2 codons, but then here's leucine down here with 4 more codons. So there are 6 codons that code for leucine. And in this second set, first letter is C, there you see it C, second letter U, and third letter U, right? Now, so in the second one, first letter C, second letter U, third letter C, and then we got CUA and CUG. So you see what happens here? This is a good example of showing you, illustrating the fact that the first and second letters do not change. It's the third letter that is variable in a codon. All right, so for all 4 of these codons, the first and second letters are CU. And the only letter that changes is the third letter, and it can be either U, C, A, or G. And so there's 4 codons here that code for leucine, and there's 2 more up there that code for leucine. All right, here's isoleucine, first letter A, second letter U, third letter U, AU, third letter C, AU, third letter A, and guess what? AU, third letter G, that's methionine. That's AUG, codes for methionine. It is also your start codon. You don't have to remember any of these designations, but you want to remember AUG as first coding for methionine, and secondly, the start codon. And by the way, this is the only codon, AUG, that codes for methionine. Methionine has only 1 codon. Valine down here has 4, you can see it. And then see, so then when you move over to the second row here, right? So in this case, first letter is A, second letter is C, C, and the third letter is either U, C, A, or G, and those are 4 codons that code for threonine. Okay, so you see how that works? And so as you're translating a message, you're going to see, a codon there, and it's going to go, and it's going to say like UCA, and so you're going to come over here and you're going to go first letter U, second letter C, third letter A. UCA is serine, so you're going to put in serine there. That's the amino acid in the polypeptide in the protein chain, right? And then you're going to say, and then you're going to read, say, UUA. Okay, so UUA. That's leucine. So you're going to put in leucine there. And let me get out of the U's. So then you're going to see GCA. And so you're going to go, first letter is G, second letter is C. That brings you down here. And you're going to go A, GCA, that's alanine. You're going to write in alanine. Okay, that's the third protein in the polypeptide. So just like that until you get to one of the stop codons, and then you can stop translating. Let's go to the video. Okay, so this is Nicole Ann's is a teacher, and she provides a really nice presentation and explanation of the genetic code. It's worth your watching, so go watch it, and the link is in the embedded videos on the embedded videos page, but watch it because it's a really good explanation of the genetic code. Okay, so now let's talk about a little bit about transfer RNA. Transfer RNA, we denote it as lowercase t, capital RNA, right? It looks kind of like this, and it's kind of all twisted together, but what we're going to do here in this presentation, we're going to untwist it and we're going to flatten it out, and that's this model right here. This is called the cloverleaf model. You can see for obvious reasons, and it's easy to see what's going on here in this presentation. Kind of hard to see what's going on there and even there, but the important thing is the 3' end is the acceptor end, and that's where the new amino acid is going to be linked to. Hold that thought. And then on the far end is the anticodon loop. All right, so this is the anticodon to the codon from the message. It'll make sense when I show you, but down here, and remember, a codon is 3 bases, 3-letter designation. So the anticodon is also going to be a 3-letter designation, but it's going to be the reverse of the codon. That's the anti part, and it'll make sense in a minute. So the loop at the bottom of the cloverleaf contains the anticodon sequence, which pairs with the codons in the message. So the amino acid that is coming in, that's going to be part of the polypeptide chain, and ultimately the protein, is attached to the 3' end. That's the free single-stranded OH at the end of the acceptor stem. You don't have to remember all those words. Just remember the 3' end up here is the acceptor end. That's where the new amino acid is going to attach to. Of course, enzymes do all of this work, and it's aminoacyl tRNA synthetase is the enzyme synthetase. It synthesizes the bond between the amino acid and the transfer RNA. You don't have to remember that, but like I said, we talked about enzymes before. You can pretty much figure out what the enzyme is doing based on its name. Okay, so here's a look at the 2D model of the transfer RNA a little bit larger, so you can kind of see the details. Now, it says over here, transfer RNA anticodon is complementary to the messenger RNA codon. So remember, here's a message down here, and here's a codon, GAG. And so the anticodon is going to be complementary to that. It's going to be the opposite, but the complement. That's the anti part, the opposite, okay? So the complement to G is C, and the complement to A is U. Remember, this is RNA, so there's uracil. And the complement to G is C. All right, so it's opposite the codon. It's the anticodon. And so what happens here is the amino acid is going to attach at this 3' end. Okay, that's the amino acid attachment site. And in order for that amino acid to become part of the growing polypeptide chain and ultimately the growing protein, the transfer RNA has to bind to the message, and it does that via the codon-anticodon complementarity, right? So if this is GAG, if this transfer was UAC, then it would not bind, right? It could not park itself on top of that GAG. So what happens is, as the ribosome moves along the message, it reads GAG, that codon. Okay. And then that transfer RNA goes to the pool of amino acids, and here we are, and it's going to look for a G, A, G, GAG, and it's going to pick up a glutamate, and it's going to bring the glutamate back and add it to the polypeptide chain. The videos will give you a great motional description of the process. Okay, so let's look at a ribosome. Now, it's the ribosomes that are reading the message. Okay, so here's the ribosome, the ribosomal complex. Here's the message. We've just cut this in half so you can see inside. Here's the messenger RNA, 5' to 3'. Okay, the ribosome is made up of two parts, a large subunit and small subunit, and I don't care that you remember which is which. Just remember it's made up of two subunits, and when those subunits come together, there are three translational sites inside, and those are important as the message gets translated. There's an A site, a P site, and an E site. And the A site stands for the amino acyl site. The amino acyl site, or the A site, we just call it the A site, binds to the transfer RNA carrying the next amino acid in line. Okay. The P site in the middle, that's the peptidal or peptidal site. It binds to the transfer RNA of the growing polypeptide. So the growing polypeptide will end up on the P site. We'll see the process in a minute. Will end up on the P site, so that the new amino acid coming in on the A site then can link up to it. And then the E site is called the exit site, and it binds to the transfer RNA that carried the previous amino acid. So as the process goes along, as the amino acid moves along the messenger RNA, right? So let's presume at the moment that we have a transfer RNA here with an amino acid on it. The next transfer RNA is going to bring the next amino acid in line here to the A site. The amino acid on this transfer to P site is going to link up to that amino acid, the new amino acid that's just been brought in. And then this now naked transfer RNA is going to move to the E site, and the transfer RNA with the two amino acids now is going to move to the P site, leaving the A site open to receive the next transfer RNA with the next amino acid. All of this is done by chemical factors that the videos will go into, and I don't want you to remember those. I don't want you to remember those fine details. But all the movement here is assisted by these binding factors, and remember GTP, the energy derived from the hydrolysis of the phosphate bonds in GTP, that energy is used to move things around. And what we'll see here in the next slide, or in the next couple of slides, is it looks like the message is moving, but the message, in this case, the message does not move to the left. The ribosome is actually moving to the right, okay? So it's actually the ribosome that's moving, all right, the message. So the ribosome here, we see small subunit, large subunit. This whole ribosomal complex is going to move to the right. So it's going to read the message, bring in the next amino acid, and then shift to the right. It's going to translocate to the right. So translation begins when the AUG, the start codon, which codes for methionine, is recognized, and the transfer RNA carrying the methionine binds at the A site. All right, that's going to be, there's your AUG, right? And initially it's at the A site. Codes for methionine. And there's a little methionine residue up there. Okay. And then the ribosome is going to shift over so that now methionine is going to be in the P lane, and the A lane, the A site, is going to be open to receive the next amino acid, which is UGG, which codes for tryptophan. That's the TRP up here. And so the new transfer RNA brings tryptophan, remember, by way of its anticodon, it binds right here. And so this whole thing binds right there, and then tryptophan is the next amino acid in the growing polypeptide chain. The methionine will then jump over and link to the tryptophan. That leaves this transfer empty, and then when the ribosome shifts again to the right, this empty transfer goes to the E site. This transfer, which now has tryptophan and methionine connected to it, moves to the P site. The A site is empty, and the next codon gets read. The next amino acid comes in. The dipeptide now clicks over. This clicks over. The transfer with the tripeptide now clicks over. The A site is empty. Okay, as the ribosome moves to the right. So, okay, that's the elongation process. So as a former transfer RNA, amino acid translocates to the P site. The next codon is read. That's the UGG here. And that transfer brings the appropriate amino acid in, which is tryptophan, and binds to the A site. Then this gets linked to that polypeptide peptide bond, and the ribosome moves again, shifts to the right. I don't know how many different illustrations that I can show you that's going to make more sense of this. When you watch the videos, I think it will become clear how the thing works. It's hard to see in these single shots, but anyway, I want to make sure you get this. Okay, so AUG here. Okay, AUG initially comes into the A site. Okay, the ribosome shifts over, and then AUG, that's the start codon, moves to the P site. That leaves the A site open. The next codon gets read, and the next amino acid is brought in by its transfer RNA and docks right there. That's what you see over here, okay? Right? Here's the octagonal or heptagonal, hexagonal amino acid. Here's the round amino acid, all right? So it's going to dock there. And then this amino acid, this hexagon, is going to be linked to this round amino acid. That leaves the transfer RNA in the P site empty, and then the ribosome moves over and everything shifts. This empty transfer moves to the exit site. This transfer now with a dipeptide on it moves to the P site, and that's what you see down here. All right, so the first amino acid gets bound to the new amino acid coming in to form the dipeptide. That leaves this guy empty. Now the ribosome shifts and this guy moves to the E site. The transfer with the dipeptide moves to the P site. That leaves the A site open. Now a new transfer can come in and bring in a new amino acid. And when that happens, then this empty transfer RNA gets booted, it gets kicked out. That's why I call it the exit site. It gets booted, and it can go and pick up another amino acid. I mean, it's not wasted, right? It gets reused. But now you have a dipeptide here, and then the next one will come in, and these two will bind to that one, and you'll have a tripeptide, and everything will shift over, and the empty transfer will be booted off. A site will be open like that. So the elongation cycle increases the length of amino acid chain. The matching transfer RNA and T codons will pair up with the appropriate codon on the message. Peptide bonds form between the amino acids. That's what you see here, peptide bonds, or peptide bond forming here. And the ribosome now moves down the message to the next codon. That's the translocation part. So it's not the message that gets pulled through like a piece of tape. It's actually the ribosome that crawls its way down the message. But I don't mean to say crawl meaning slow, because it's not a slow process at all. Okay. So the elongation process is when you connect the first amino acid to the new amino acid, making a dipeptide, and then the dipeptide gets connected to the third amino acid, that's a tripeptide, and to the fourth amino acid, that's a polypeptide, to the fifth one, and it grows and grows to however many amino acids you have in that chain or in that message. And this is just showing you that elongation process. So as this guy comes in with this amino acid in green, this whole polypeptide chain, this amino acid chain, will then connect to that guy, leaving this transfer open, and then when the ribosome shifts, this empty transfer goes over here to the exit site. The new transfer with the elongated chain goes over here to the P site, and that leaves the A site open for the next amino acid. Termination of translation happens when you get to a stop codon. Remember, AUG is a start codon, and so you read AUG, that tells the ribosome start translating AUG, AAU, GGCs, CAU, like that all the way down, and then you get to a stop codon, UAA, UGA, or UAG, and then when the ribosome gets to a stop codon, then it tells the ribosome, then the stop codon tells the ribosome, okay, you can stop translating. The polypeptide chain of 100 amino acids or 1,000 or whatever it is gets clipped off, and that is the amino acid chain. That is the polypeptide. That is the linear protein that then is going to get folded up and is going to be useful. So stop codons are recognized by release factors, which release the polypeptide from the ribosome. That's the polypeptide chain, the amino acid chain. You see it here. And that's the linear protein. Okay, video from Student Help for AP. This is translation, elongation, and termination. Pretty good video, and it explains, it kind of goes fast, but you can watch it multiple times. but you'll see how that process of the growing polypeptide chain, how the chain elongates, and then finally how translation gets terminated. Okay. Now, what is the difference between prokaryotes and eukaryotes? Well, there's a number of differences. For one thing, in the prokaryotic message, there are no introns, so there is no pre-translation modification that needs to be made where introns are clipped out and exons are spliced together. Okay. The number of genes in the message says several genes may be transcribed into a single messenger RNA. Now the thing about prokaryotes is it's a simpler system, and so there is a lot going on at once. With eukaryotes, we transcribe the message, and then once that message gets transcribed, it gets processed, and then it gets sent out of the nucleus, and then it gets translated like that. With prokaryotes, the message gets transcribed from the DNA, and then once that message gets transcribed, then lots of ribosomes jump onto that message, and so you have multiple translations going on at the same time. So because there's no membrane-bound nucleus, transcription and translation are coupled. So on the one hand, you can have transcription going on, and even as transcription is happening, once you get a certain number of bases in the message, then that message can start being translated. So even though the message is not completely transcribed, you can begin to translate it as a bacterium. Right? So remember, with eukaryotes, right, we transcribe the message, we process the message, we send it out of the nucleus, then we translate the message. With prokaryotes, transcription can begin, and while transcription is happening, translation can begin, and translation can happen at multiple sites. So there's a lot going on there because there's no nuclear membrane, and there's no pre-translational modifications that need to happen. So down here it says modifications of message after transcription. None. There is no modification, but there are a number of modifications that eukaryotic messages have to go through before they can get translated. Okay, so here's a video by Alila Medical Media of the translation process in prokaryotes. Okay, and it's pretty good. It's very simple, and it's at a sort of a kind of an easy pace. It's easy to swallow. Of course, you can pause it at any moment. But it shows you protein synthesis or translation in prokaryotes. It's a pretty good little video to watch. And so to wrap this up, here you see, this is kind of cool, and I'll direct your attention to this in a minute. Prokaryotic transcription is coupled to translation. In other words, the message or RNA begins to be translated even before transcription is complete. Remember that eukaryotic transcription occurs in the nucleus. It has to be the message has to be modified. It has to leave the nucleus, and then it can get translated in the cytoplasm. This micrograph here shows you many points of translation happening even before the message is completely transcribed. So here you see the DNA strand up here. That's what you see in illustration down here. Here's the DNA strand. Okay, here is the messenger RNA, and all of these little gold dots represent ribosomes, and all of these little hairs coming off, those are proteins. So you can see this message It hasn't even been fully transcribed yet. It's still coming off of the DNA. But even before it's fully transcribed, these ribosomes are jumping on there and they're translating. And that's what you see here. These are the little polypeptide chains. These are the amino acid chains. These are the little primary structured proteins that are happening here even before the thing is completely transcribed. Okay, pretty cool how that's going on. So all of these little dots here that you see, these little dark dots, those are ribosomes doing their work. These are all ribosomes that are jumping on these little messages, and they begin translation even before transcription is complete. Pretty cool how that works. Preguntas, if you have any questions, message me, email me. You can contact me through the Canvas inbox, and I will see you in the next round.